Here we have the mechanisms of the hydrolysis of acetals, imines, and enamines from the aldehyde and ketone chapter, which is chapter 19 in Klein. And this is something I prepared for the winter class, and I went over with them, and now I'm going to go over it for the spring class. The Klein textbook goes through the mechanism for the acetals, but not for the other two. So this is an opportunity to look at all three mechanisms and see their similarities and differences. All three of these mechanisms are shown in acid, which means that all of the intermediates need to either be acidic or neutral. Remember that you cannot generate strong bases like alkoxide anions in acid, just like you can't generate strong acids in base. So always keep that in mind. Another thing that sometimes helps students with these mechanisms, kind of depends on how your brain works, but some people kind of think about it as what happens in each case. So for example, with this first one, you have a proton transfer, leaving group, nucleophilic attack, proton transfer, leaving group leaves, proton transfer. And some people can kind of, think of them that way. If nothing else, it does remind you that there are a lot of proton transfers in these things. And in general, if you ask yourself, do I need to do this with one proton transfer or several, the answer is usually several because the protons aren't going to hop. So let's start with the acetal. So the acetal is the one where you have a carbon that's attached to two oxygens. So that's what I sometimes call a full acetal. And it's helpful to be able to recognize that. So the first thing that has to happen is we need to have this group leave. So in order to make that into a good leaving group in acid, we protonate it. That gives us this intermediate. The leaving group now leaves, and unlike a regular SN1 reaction, when the leaving group leaves, in this case we have another oxygen with a lone pair that can move in and stabilize that carbocation. Sometimes you'll see this written as two steps, so you'll see the leaving group leave and then the resonance. So that gives us this species, and the oxygen nucleophile now attacks the carbonyl carbon, the electrons move out, we end up with an oxonium ion, and you can either have something that's a strong acid dissociate on its own, or you can pull it off with a suitable base, like water in this particular instance. Once we get to here, we've now reached the hemiacetal. Now we need to get rid of this group, and so we're going to protonate it, make it into a better leaving group, and then very similar to this step where the leaving group left, this leaving group leaves in much the same way, and the lone pair on the oxygen moves in. Now we have the conjugate acid of the carbonyl compound, and again, this can either protonate on its own, or you can proton transfer it with something else. You may or may not remember from your general chem this concept of the principle of microscopic reversibility, which says that if a reaction goes forward by a series of steps, the reverse is the same but backwards. And that certainly works in this case. If you look at these steps backwards, it's the steps to take the carbonyl compound and make the acetal out of it. And again, the carbonyl, or the former carbonyl, or the carbonyl in the acetal is recognized by having these two oxygens. So in all three of these hydrolyses reactions, we're going to be breaking bonds to heteroatoms, because the heteroatoms that are attached to this carbon ultimately become the leaving groups. So now moving on to the imine, which is the result of taking a primary amine or NH3, in other words, something where the nitrogen has at least two hydrogens on it. So this imine And again, the first step is a proton transfer onto the nitrogen. Now the water attacks not the nitrogen carbon, or not the nitrogen, but the carbon next to the nitrogen, which is pretty similar to what happened in the above case. That gives us this intermediate, which is the conjugate acid of the so-called carbanolamine. And this carbanolamine is analogous to the hemiacetal. So you have an OH and a nitrogen with one hydrogen and other groups attached to it. So now we need to get rid of this nitrogen group, and we're going to do it by making it a better leaving group, by doing a proton transfer. And now in a step that should look pretty familiar, the leaving group leaves, the lone pair on oxygen moves in, and we get, again, the conjugate acid of the carbonyl compound. In this case, though, what happens is that this compound, which is a strong acid, reacts irreversibly with the amine and protonates the amine to the ammonium compound. And that helps this reaction to occur because once you've generated the protonated form of the amine, it can't go backwards. And that's why I showed that with a one-way arrow. If you compare this to the mechanism for taking a carbonyl compound and making an amine, it is different in that that reaction doesn't have this intermediate. Instead, what happens is the amine attacks the carbonyl compound, and instead of a protonated oxygen, you end up with an O- in the forward reaction from the carbonyl. But that reaction is done at slightly higher pHs, still acidic, but more like pH 4 to 5, whereas this would be done in lower pH to drive the reaction to completion. So finally, we can tackle the enamine. The enamines result from having reaction between a carbonyl compound and secondary amine. So in other words, the original amine has one hydrogen on nitrogen. So again, we start with a proton transfer. This step is kind of more analogous to the first step of an electrophilic addition. And the hydrogen goes on the carbon that's one away from the nitrogen, because by doing that, you can generate a carbocation here that can be stabilized by the lone pair on nitrogen. So all three of these mechanisms have a lot of similarities to SN1 and here E1 reactions, or not E1, but addition reactions, except that because we have this heteroatom involved, it looks kind of different. So again, the nucleophile attacks the carbon that's next to the nitrogen. We make the conjugate acid of the carbanolamine. We deprotonate that to make the carbanolamine itself. And then just as happened up here, the amine group has to leave the carbanolamine by first getting protonated. Then the leaving group leaves. We form the conjugate acid of the carbonyl. And again, we do the same proton transfer that helps to drive this reaction to completion. So again, this reaction, the hydrolysis of the enamine, is the exact reverse of making the enamine, except that, again, we generate this protonated carbonyl intermediate that's not a factor when we go in this direction at a slightly higher pH. One interesting thing you can do is you can do these reactions kind of side by side or one right under the other one. But if you look, for example, at the imine versus the enamine, the first step happens differently. In the case of the enamine, you add hydrogen to a carbon to generate this intermediate. Here, you protonate a nitrogen to generate this intermediate. But you can see that these two intermediates are very, very similar. And all of the subsequent steps, once you get to this intermediate, are exactly the same for both the imine hydrolysis and the enamine hydrolysis. The acetal hydrolysis is a little bit different, but again, it might be instructive to compare and contrast the steps here written right above the steps for these two. Generally, these two reactions the hydrolysis of the nitrogen compounds seem less intuitive to a lot of students. So it does help to recognize the similarities between the two of them. And again, if you look at them from the standpoint of what's happening, some people find this very, very helpful. Do it in a slightly different color. So we have a proton transfer, nucleophilic attack, proton transfer, leaving group, leaves and resonance, proton transfer. And those would be the exact same steps down here. And if we can also compare it to that, and they're not quite the same there. So that's this